Hello, brave biology students. Today I'm going to go over the mitosis lab, starting on page 44, the results section. You guys did a good job finding the different phases, and you might not have the exact same pictures on the exact same page. If you would have started with perhaps the Horosiscaris, you might have put them on page 44, and you might have put a different phase. This one starts with prophase and ends with interphase. You might have started with interphase. So just be careful when you look at yours to make sure you're identifying them properly. This is an example of the onion root tip in prophase. Ours were a lot rounder than this. It's got the nucleus identified here. Yours might be a rounder cell. If I would have drawn it, I would have drawn a more round cell. And then it's got the magnification. The next one it did on page 44 is metaphase. So here's a good look at metaphase. Yours might not look identical to this. It's kind of got the chromatids lining up in the middle and the spindles forming. Anaphase is the next one they did on this one. And again, it's separating out more. So you might have a cell that's just elongated and it looks like it's dotted all the way through. That would also be a good anaphase. Page 45, it continues here with the next one they did was the onion cell telophase. Telophase, it's getting ready to have that daughter cell. In the plant cells, it's not called a daughter cell. It's just put the cell plate in the middle, but it looks like a daughter cell. And again, ours are probably rounder, and it shows the two nuclei. And then it shows its last phase, it shows interphase, which is pretty much a regular cell that's not dividing. It's in between division. On 45, number 2, they start with the Horosiscaris, and they start with prophase. Okay, so you might have a different phase here. Prophase for here is it's just starting to expand out. Maybe the nucleus is bigger in your cell. The next one they have is metaphase, where it shows the chromatids coming apart. There was a pretty good identification on Sam's microscope of this, where it actually showed the little lines of chromatids and they were separating. Anaphase, they're more separate and it's elongating more, and you might be able to see spindles, the little dotty things. When it gets to telophase, you've got those daughter cells. And then again, it ends with interphase, which is pretty much a regular cell before it starts to divide. That was page 46. Again, if yours are in a different order or a different type of cell, that's perfectly fine. Oop, wrong page. All right, on page 48, starting with the questions, we'll answer those now. Question number 3. List and describe the stages of mitosis in an animal cell, starting with interphase. You may or may not have these same wording in here, but there's some key phrases in here for you that you can jot down if you don't have this one completed. Starting with interphase A, that's the growth phase of your cell. They're not dividing during interphase. They're replicating the DNA in organelles, and the cell is growing. Prophase, which is B. There'll be changes in the nucleus and the cytoplasm of the cell. In the nucleus, the chromosomes become visible. They shorten and thicken up. They coil up. They sometimes appear as two chromatids connected by a centromere. Kind of in the middle, they're connected. Nuclear membrane and nucleolus disappear. And in the cytoplasm, the centrosomes migrate to the opposite poles of the nucleus, and that forms your spindle fibers. And those are those dotty-looking things that we saw. Metaphase, the centromeres align on the equatorial plane, so they're in the middle. They look like a little line or a little fence. Spindle fibers attach from each new daughter cell pole and to each chromosome at the centromere. So they're going to attach those and they're going to pull those apart into each of the cells. D is anaphase. The chromatids separate. Chromosomes pull toward opposite poles of the cell. And finally in telophase, I'm going to move this up so you can see it. The chromatids reach the end of the spindle. The chromosomes begin to uncoil. The nuclear membrane and nucleoli reappears. Cell membrane pinches in half to form the two new daughter cells, or the cell wall grows up in between the two daughter cells in a plant cell. Number 4. Why is mitosis essential to the life of organisms? Well, mitosis is cell division. And without cell division, we don't have duplication of cells for growth, and we don't have replacement of damaged cells. So it's for growth and for replacement of dead and damaged cells. Number 5, were you able to see all the same phases of mitosis in the plant cell as well? Were you able to see all the phases? I think we did a pretty good job seeing all the phases. You might not have seen, they might not have been completely clear in the plant and animal cells, but I think we took a good job. The answers are going to vary on that. I would say yes, we saw a lot of it. Number 6, during mitosis, what structures are present in animal cells that are not found in plant cells? The structures in animal cells but not plant cells are your centrosome, your centrioles, and your aster. During telophase, the daughter cells are pinched off in an animal cells, whereas in a plant cell, they're separated by a cell plate, which forms a cell wall. During mitosis, what structure? You're going to have to find that in your textbook. That's not in your lab manual. During mitosis, what structures are the same in both? Both have chromosomes and both have spindle fibers. Number 8, how does separation of daughter cells differ in plant and animal cells? We already talked about that. Plant cells form the cell wall between the daughter cells, and animal cells pinch in two. How do the poles of the spindle differ in animal cells and in the plant? In animal cells, a centriole is present at the poles of the spindle. Again, that's like a textbook question. So that was actually covered pages 47 and 48 for the questions in your lab manual. Good job on that lab. We got a lot done. For homework, I'm going to ask you to read both bacteria labs, Bacteria 1, we're going to do in class with the prepared bacteria slides, and Bacteria 2, you're going to grow some mouth bacteria at home during next week. So have a great rest of the week, and I'll see you on Monday.